science explorer. So, have you ever walked with your friend and suddenly noticed you're going in opposite directions? It's a little weird, right? But in the world of DNA, that's actually a smart design. By the end of this lesson, students will be able to define the term antiparallel in the context of DNA's double helix structure, describe the directional orientation of DNA strands as 5' to 3' and 3' to 5', explain the significance of the antiparallel arrangement in base pairing and DNA stability, illustrate the antiparallel nature of DNA using diagrams or models, and discuss the role of antiparallel strands in the process of DNA replication. So, DNA is made of two strands, and they don't run in the same direction. So one goes from left to right, and the other one goes from right to left. This is what we call antiparallel, and it helps DNA store, pair, and copy your genetic information correctly. So in this short lesson, we'll talk about what antiparallel means, why it's important, and how it helps during DNA replication. So now, let's dive in. Before we begin, take a moment to really look at this animation of the DNA. Familiar? Probably, but here's something to think about. What do you remember about the DNA structure? And why do you think that its two strands run in opposite directions? So what does antiparallel mean? When we say the DNA is antiparallel, we mean that the two strands in its double helix run in opposite directions. So each strand has a direction based on its sugar phosphate backbone. One end is the 5' and the other end is the 3'. In the DNA double helix, one strand goes from 5' to 3' and the other one goes from 3' to 5'. Think of it like two lanes on a highway, traffic flowing smoothly, but in the opposite directions. And that's not just a structural detail, it's essential. This opposite direction allows for complementary base pairing. So adenine pairs with thymine and cytosine pairs with guanine. That pairing is only possible because the two strands align in opposite ways. They're like puzzle pieces that only fit when flipped the right way. Now try this out yourself. Open your digital activity template and complete the drag and drop task. What you have to do is to match the complementary base pairs and label the direction of the second strand from 3' to 5' based on a given 5' to 3' strand. Now pause the video and resume once you're done. Take your time! Great job! Now let's take a step deeper. So here's a video to further understand its structure.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated.
But there's more. When it's time for the DNA to copy itself, during replication, an enzyme called DNA polymerase comes in. It's the builder. But here's the twist. DNA polymerase can only add nucleotides in one direction, from 5' to 3'. So how does it work on two strands facing opposite ways? One strand is easy. It's copied smoothly and continuously. So that's what we call the leading strand. But the other strand is more complicated. it gets copied in small chunks called the Okazaki fragments. That's the lagging strand. So this difference happens because of the anti-parallel orientation. So let's go back to the video.
The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again.
Picture this. The DNA is about to be copied inside the cell. One strand is like a straight open road, easy and fast to replicate, and the other is more like a zigzag. Stop, build a piece, jump back, and build again. Now that's the real world effect of being anti-parallel. It's like building a road in reverse but with perfect precision. DNA structure ensures that even with complexity, replication stays accurate with efficiency. Now it's your turn to apply what you've learned. Complete the provided worksheet. 1. Fill in the complementary strand for a given DNA sequence. 2. Label the strand from 3' to 5'. And 3. Which strand do you think would be the leading strand during replication? Now use this activity to lock in with what you've just learned. DNA strands are anti-parallel. One runs from 5' to 3' and the other from 3' to 5'. This orientation ensures complementary base pairing and allows DNA polymerase to do its job. Because of this structure, DNA replication is precise, with leading and lagging strands working together in perfect balance. Anti-parallel strands aren't just a detail in DNA's design. They are the foundation of how life replicates itself accurately from generation after generation. Let's wrap things up and check your understanding. Pause the video and write down your answers to these questions. First, what does anti-parallel mean in DNA? Second, in which direction does DNA polymerase work? Third, why is strand direction important in replication? And finally, reflect on this in just one sentence. How does having DNA strands in opposite directions help keep our genetic code accurate? Now take a moment. Your answer might be short, but the concept is also powerful. opposite directions is what holds everything together.